The world today is experiencing the largest global health crisis in the last century. It is a time of uncertainty and fear for many people. COVID-19 is spreading throughout the world, causing infections, quarantines, stay-at-home orders, travel restrictions, and untimely death. In this time of uncertainty, we can look to science for an explanation and hope for the future. Luckily, we are not alone. Researchers worldwide are conducting experiments, testing potential treatments, working on vaccines, and trying to help the world recover. This project will exclusively use and cite research from outside of the United States. Before we look at how the world is treating COVID-19, let's look at what it is and how it works. Firstly, COVID-19 is not the name of the virus, but the disease caused by it. The official name is Severe Acute Respiratory Syndrome Coronavirus 2, or simply SARS-CoV-2. After its initial emergence in Wuhan, China, researchers immediately began investigating everything about the virus. Its viral genome was successfully sequenced just 2 weeks after the first infection on January 10, 2020, by the Shanghai Public Health Clinical Center. This led to further discoveries about its characteristics. Using transmission electron microscopy imaging, the virus was shown to be spherical in shape with distinctive spikes about 8 to 12 nanometers in length. This was also the imaging that produced the iconic photo that appears on everyone's nightly news. It was determined to officially be a part of the coronavirus family due to these discoveries. It is also important to understand how the virus was transferred to humans. Multiple studies using the full genome of SARS-CoV-2 have found that it is very closely related to many SARS-related coronaviruses in bats. At the genomic level, SARS-CoV-2 has 96% sequence identity when compared to a bat coronavirus, according to Chinese scientist Zhou et al. This level of similarity indicates the presence of an intermediate carrier that passed it on to humans. By looking at the proposed phylogenetic tree by Chinese scientist Li et al., it is clear how closely related the sequences of human, bat, and pangolin SARS-CoV-2 is. This shows a possible connection about the transmission of the virus from species to species. This phylogenetic tree also emphasizes how similar SARS-CoV-2 is to SARS-CoV. This evolutionary relationship is very important to consider because it can help with the development of treatments and overall understanding of how the virus functions. So how does it function on an individual and cellular level? What is the path of infection? Firstly, the virus is airborne and is transferred by human contact, hence the need for worldwide isolation. Let's follow the path from Jane, an infected individual, to Joe, a healthy individual. Joe and Jane are talking less than 6 feet apart, and Jane's breath is spewing the virus into the air. The virus is landing on Joe's skin, clothes, and potentially in his mouth when he opens it to talk. As the virus travels inside of Joe's body, its protein spikes are looking for specific receptors to latch onto, kind of how keys only work in specific locks. The locks for the virus are angiotensin converting enzyme 2 or ACE2 receptors. In a healthy body, ACE2 receptors are part of the renin-angiotensin system that regulates blood pressure and fluid and electrolyte balance. After the spike proteins connect to the ACE2 receptors, the host cell allows the virus to enter. The viral envelope comes apart, releasing viral RNA into the cell. The viral RNA is picked up by the ribosomes and starts to synthesize polypeptide chains. These polypeptide chains produce more copies of the virus inside of the cell. Based on the sequencing of the genome, it is known that 1/3 of it encodes 4 main structural proteins: spike, envelope, nucleocapsid, and membrane proteins. The other 2/3 are 2 large polyproteins that form the viral replicase transcriptase complex that replicates the viral RNA to pass on in future virions. After the viral genome is replicated, the envelope glycoproteins are inserted into the ER or Golgi and form a nucleocapsid. Then, they move to the ER-Golgi intermediate compartment. The formed vesicles move to the membrane of the cell to be released by exocytosis. These copies are released into the body and start to look for more ACE2 receptors to bind to and host cells to infect. This is how the virus spreads throughout the body. Now that the virus is in the body, how does it cause symptoms? Because of its complexity and the lack of time for thorough study, most explanations are hypotheses that use the data recently collected to try and explain how it affects the body. This is in no way a complete explanation, but an attempt to outline some of the proposed hypotheses and how they affect the body. When the body is infected by a virus, innate immunological responses are activated to stop the spread and mitigate the effects. Pathogen-associated molecular patterns, or PAMPs, are the systems set up in the body to recognize the viral genome and initiate signaling pathways and transcription factors to slow or stop the spread of the virus by not allowing it to be made in the cell. One of the most well-known and effective immunological responses to viral infection is type 1 IFN or interferon response. These are polypeptides secreted by infected cells that work to reduce infection in 3 ways. 1. It induces cell-intrinsic antimicrobial states in infected and neighboring cells. This means that the cells are less likely to uptake anything from the exterior and use its foreign RNA. This limits the spread. 2. It modulates immune responses by promoting antigens and natural killer cell functions. This is how infected cells can get rid of the virus and healthy cells recognize not to allow the virus to replicate. 3. It activates the adaptive immune system with antigen-specific T and B cell responses and immunological memory. This makes the virus identifiable and allows the body to remember it in the future. The type 1 IFN response is extremely important to the immunological response to viral infections. According to a recent study from Iran by Rokni et al., SARS-CoV-2 suppresses this response, making it harder for the body to fight the infection. In general, T helper cells are used in the immunological response to identify antigens and secrete cytokines to alert T and B cells that they have located the virus. Th1 and Th17 are 2 T helper cells that secrete pro-inflammatory cytokines like IFN gamma. These pro-inflammatory cytokines cause a chemotaxis in cells and attracts neutrophils and macrophages. Neutrophils are white blood cells that create inflammation as their immunological response. Normally, the type 1 IFN response acts as a control to this effect, but while it is suppressed, it can do nothing to stop this from occurring. This inflammation gathers in the cells of the lungs, creating fluid-filled air sacs. This hypothesis concerns how pneumonia develops as a result of SARS-CoV-2. Another study from China by Li et al. Agrees with this hypothesis and postulates that the cytokine storm of large amounts of pro-inflammatory cytokines and chemokines by immune effector cells leads to an uncontrolled systemic inflammatory response. This leads to acute respiratory distress syndrome, or ARDS, the main cause of death due to COVID-19. Another cause of symptoms has to do with the blocking of ACE2 receptors by the virus. As mentioned before, this is how SARS-CoV-2 enters the cell. In addition to that, the virus also dampens the functions of ACE2 and the renin-angiotensin system. This system is responsible for the regulation of blood pressure and fluid and electrolyte balance. ACE2 acts as a negative control for ACE by reducing inflammation, fibrosis, and atrophy. It promotes tissue protection. A study from Austria by Imai et al. Details how the dampening of ACE2 leads to acute lung injury and eventually acute respiratory distress syndrome, or ARDS. While studying mice, they discovered that acute lung injury was only associated with decreased ACE2 expression and an increased production of angiotensin 1. The balance of the renin-angiotensin system is lost. ACE promotes disease pathogenesis and injury, while ACE2 alleviates it. Loss of ACE2 function leads to increased vascular permeability that leads to pulmonary edema, which is a key characteristic of ARDS. Now that we have the basics of how host cells are infected and some of the influences on the immune system, we can move on to the hopeful side, a treatment. An understanding of the virus is necessary before treatment and vaccine development can begin. As we continue to learn more, we have more options for treatment. Let's take a look at hydroxychloroquine, the medication that has been widely publicized as a potential treatment for COVID-19. How would it work and is it effective? One study by China by Zhou et al. Investigates hydroxychloroquine and how it would inhibit SARS-CoV-2. As a disease-modifying anti-rheumatic drug, hydroxychloroquine has been used for treating rheumatic disease and preventing inflammation and organ damage. Hydroxychloroquine acts by increasing intracellular pH and inhibiting lysosomal activity in antigen-presenting cells. This prevents antigen processing and reduces T cell activation and cytokine production. As discussed previously, one of the main causes for ARDS and severe symptoms is the cytokine storm promoted by the virus. Hydroxychloroquine also disrupts the transcription of pro-inflammatory genes. It could block the invasion and replication of the virus and assuage the cytokine storm. Because hydroxychloroquine has already been in use since World War I and is commonly taken for malaria and inflammation, it is well known and studied. This makes it an excellent candidate to be tested for its effectiveness in COVID-19. So now that we know how hydroxychloroquine works, how effective is it? One study in France by Gautret et al. Used a small sample size to test the effectiveness of hydroxychloroquine on respiratory viral loads. The 20 patients tested had a significant reduction in their viral respiratory loads and much lower average carrying duration than untreated patients as reported in the scientific literature. They concluded their results displayed enough significance. Another study in China by Chen et al. Had a similar sample size of 30, and while there was a reduction in viral respiratory loads, it was not significant enough. Other factors like time for the fevers to subside and improvement in follow-up appointments were very similar and showed no significant improvement with hydroxychloroquine. These studies are examples of the importance of large sample size and continued testing to determine the effectiveness of medicines. Based on these findings, there may be promise for hydroxychloroquine, but there is much research yet to be done. Let's go back to viral entry into the cell through ACE2 receptors. Even in just this aspect, there are 4 potential therapeutic approaches as referenced by a study out of Ontario, Canada by Zhang et al. The first approach is a SARS-CoV-2 spike protein-based vaccine that would promote the production of antibodies, recognizing the spike and making the body more prepared for a potential infection. The second approach is a transmembrane protease serine 2 or TMPRSS2 inhibitor. TMPRSS2 is used by SARS-CoV-2 in priming the spike protein for attachment to host cells. By preventing this priming using an inhibitor, the spike would be unable to bind to host cells. The third approach blocks the surface ACE2 receptors by using anti-ACE2 antibodies or peptides. These antibodies or peptides would attach to ACE2, blocking SARS-CoV-2 from binding. The fourth approach is delivering an excessive soluble form of ACE2. This is like providing the virus with decoy cells so that it is unable to replicate as easily in host cells. COVID-19 spread fast and hit hard. Our daily lives have been radically changed to adjust to the new world that we live in. While unexpected, this pandemic will not last forever. It is comforting to note that researchers globally are not stopping in their quest to better understand and treat the virus. We are not alone. Stay home and stay healthy.